Well, thank you very much. Thank you everyone for the kind invitation and for the very interesting webinar today. My job will be to talk a little bit about immunotherapy to treat hepatocellular carcinoma, which has been, a topic very close to my heart, very close to my own even, career development. So I'm actually very enthusiastic to talk to you about one of the novel avenues to treat what we all know has traditionally been a highly lethal tumor. And I'll try and navigate very quickly through my slides to keep it interesting, to keep it lively, maybe not present all of them. So I think it's fair to say that we can't talk about immunotherapy without also reflecting about what else is there, what is the real role of systemic therapy in HCC. And as a medical oncologist, I'm not ashamed to say that our role in treating HCC has been comparatively smaller compared to many other disciplines up until quite recently. And I will explain why that is, purely because there haven't really been a lot of agents capable of inducing responses and changing the long-term prognosis of these patients. I will try to convince you that immunotherapy can do that, both from a perspective of the mechanism of action, but also from the perspective of the clinical data that are supporting the use of immunotherapy. And obviously we can close, as a joint reflection in this very interactive webinar, trying to think about what are the open questions and potentially the new challenges that we have to think about when we use these novel and highly effective treatment options in patients with HCC. It really depends if you're an optimist or a pessimist. You can either see the world of systemic therapy in HCC as a glass that is half full or half empty. But surely, if you think about the beginning of the story, 60s, 70s, 80s, where in other tumor types, we were starting to have initial experience, with cytotoxic chemotherapy leading to really impressive survival advantage in lung, ovarian, breast cancer. You can see that those decades were really left to neglect for HCC. We have to wait 1999 to agree on some form of staging, which still to this day is a little bit debated. Do we like Barcelona? Do we not like Barcelona? Still, open to questions. And then another decade to think about sorafenib as the first drug, interrupted by a number of late-stage failures. A lot of us have been investigators of these trials, a lot of TKIs that never really made it to the market, both in advanced disease and in adjuvant and in combination with TACE. And then you look at the right-hand side of this trajectory, 2023, I need to update it to the current year, where I'm sure there will be many more drugs coming through. Truly an explosion of novel TKIs, VEGF inhibitors, but particularly immunotherapy. It's fair to say, though, that if you think about TKIs, which have been the standard of care for over 10 years, there are a couple of issues that you will be finding immediately evident from these curves. The first one is that the median overall survival from treating patients with HCC on TKIs is actually quite low. So in the original studies, we're really thinking about a median OS gain of about a couple of months. And even though novel TKIs, such as lenvatinib, for instance, have been shown to be slightly better, for instance, although formally never superior, but simply not inferior to sorafenib on the basis of the REFLECT study, you can see that maybe the response rate is a little bit better, no longer 2%, but maybe around 20%. But the thing is that with these drugs, you really hit a brick wall at about 1 year survival, and patients don't really do any better. The other thing is that we don't really have predictive biomarkers. So a lot of people that treat, for example, colorectal cancer, will be aware of KRAS, unlike non-small cell lung cancer, melanoma, etc. We don't really have a molecular guide. Tolerability is an issue. And as we said, not much beyond the 1 year survival landmark for these patients, although there have been some interesting changes in the recent past. What has really changed the perspective, and I'm glad that, we mentioned people like Alexander Knuth earlier on, who was one of my earlier mentors in my pathway of medical oncologist, is really the early work of immunotherapists. And Alexander is not as old as Professor Coley, who basically in 1908 was probably the first one to really think about immunotherapy by injecting bacterial toxins. But him and many other people, looked more into how to make this process more selective. And I think traditionally we are used to think about vaccines, cytokine therapies, which although made a really good contribution to the understanding of the cancer immune synapse, actually, as therapies, these treatments were very, very disappointing from the point of view of leading to important changes in management. The tipping point is the study by Hody, published in 2010. That molecule over there, a rather obscure monoclonal antibody that many of us didn't really understand even how it worked, ipilimumab, was actually characterized in a disease that you will think is lethal in about 3 to 6 months, metastatic melanoma. Well, that molecule was actually capable of shrinking the disease in about 11% of the patients, but actually achieve that long-term control that you can see in those little red arrows there, which is really what makes immunotherapy quite fascinating as a treatment modality. The fact that some people remain with their disease completely static without need for any treatment, and they actually survive up to remarkable landmark endpoints, 40, 50, even 56 months, as you can see from the Kaplan-Meier figure. It's no mystery that we have 2 main actionable drivers of anti-cancer immunity, programmed cell death 1 and CTLA-4. So these are 2 different pathways that regulate T-cell exhaustion. PD-1 probably more at the peripheral level, at the interface between the tumor cell and the T lymphocyte, the cytotoxic CD8 positive T cell, and then the CTLA-4 molecule, which is more relating to the way those effector T cells are primed at the level of the lymph node, at the level of the antigen-presenting cell. The latter part of my slide really documents how these novel treatments have expanded since the early days until now and even beyond, with a number of indications that have been really changed in the way they are managed by the introduction of immunotherapy, either alone or in combination with chemo. And so you will think, what has happened in HCC? And we've studied HCC for years, and we have plenty of tissue that we profiled, and we now know, thanks to the work of plenty of investigators, that there is a particular subset of tumors that are characterized by some form of spontaneous immunogenicity. So what we call the immune class of HCC, some of them have a presence of active immunity, so evidence of active killing of cells by cytotoxic T cells. And you can see all the traits, the presence of high PD-L1 expression, immune-related genes that suggest spontaneous immune activation. And on the right-hand side, you can see interesting Kaplan-Meier curves that suggest that this phenotype really leads to a difference in terms of outcome for these patients. So that is good enough to say, well, there is a case maybe for immunotherapy in HCC. However, what you then end up seeing is that the forerunner molecules, PD-1 inhibitors, nivolumab and pembrolizumab, in these 4 clinical trials that were meant to change the landscape of HCC forever, actually they all failed miserably. Some of them even against placebo. Look at the Keynote 240 trial, where despite a potentially positive p-value, actually that p-value is still negative because of the way the statistical analysis plan was devised. And so you are left with these Kaplan-Meier curves, which give rise to the most significant median overall survival ever seen in first line before of 16.4 months and even 13.9 months in second line. You think, wow, these drugs are really working, but then they're not. The p-values are negative and there is no evidence of a survival benefit, leading to these drugs to be forgotten as a potential treatment change in these patients. And there are a number of reasons why PD-1 monotherapy failed. First of all, the patients were unselected, so we don't know which patients were treated from a molecular perspective. The study design was actually quite ambitious. A lot of these companies wanted to place their molecules quick and easy, aiming at superiority over standard of care. The comparator arms were tricky because, for instance, people in the United States could access PD-1 after progressing on sorafenib standard of care, and therefore this must have muddied the water with crossover post-PD progression disease therapy. And also, we have to be honest, PD-1 inhibition in HCC is effective, but limited effectiveness, probably because of this complex scenario of immune suppressive cells. You can see at the bottom of the slide an exhausted CD8 positive T cells, which is fully charged of PD-1, CTLA-4, but they can't really overcome a number of other negative regulators within the microenvironment that are coming from a number of cells, myeloid-derived suppressor cells, exhausted CD4 T cells, NK cells, Tregs, etc. So it is a bit too much to ask for a tiny little thing like an exhausted T cells to basically have to fight the corner against all these immunosuppressive cells. And we do really have to think about what else to do to combine with PD-1 to enrich the responses in these patients. And I think, those failures led us to go back to the drawing board, really. And I think there isn't more of an eloquent drawing board for myself. I'm not an immunologist, but I'm always fascinated about the fact that we do have almost like an ingredient book for cancer immunotherapy. And we should always remember this to heart, because for us to be able to use immunotherapy wisely, we need to have all these 7 ingredients, the 7 spices of immunotherapy, to be active and to be present in our microenvironment. We need good antigen presentation, activation, trafficking of T cells, and then the killing. And it's fair to say that PD-1 monotherapy only really tackles the recognition and killing. It doesn't tackle all the other aspects of the cancer immunity cycle, which is why perhaps, PD-1 monotherapy was not effective in HCC. We have realized this, and what we really want to do is to turn the page and think about combination therapy. We think it's very easy. It's almost like, an easy treatment. Just combine more things and you'll be fine. But the problem is that combining is not as easy, because you can see on the right-hand side the explosion of combination studies with absolutely everything under the sun. All these little bubbles are basically telling us what is being combined. And in HCC particularly, the combination with other second checkpoint inhibitors, VEGF inhibitors, TKI, has been the cornerstone of this second wave of trials. But I must say that not all of them were positive, and we'll have to still do a lot of work to understand why we are still hitting a lot of failures. A lot of failures, despite what I think is a very convincing background to these therapies, in particular when it comes to VEGF. The reason why VEGF is particularly interesting is because we always felt that VEGF was this key cytokine. At least, I learned it when I was studying medical oncologist as a registrar, that it's this important molecule that governs the process of production of new blood vessels that are feeding the tumor. This is certainly true, but VEGF is also an immunomodulatory cytokine that is involved in T cell trafficking and infiltration, that is involved in antigen presentation and T cell priming. And guess what? PD-L1 expression in the tumor cells is very much driven by HIF-1 and HIF-2 alpha, which are again hypoxia sensitive elements. So the more hypoxic and the more VEGF stimulated the tumor is, the more there is presence of PD-L1 and therefore immune exhaustion. So there is a very strong rationale to combine both with TKIs and with VEGF inhibitors. And you will see that this particular idea has actually translated very well in what has now become the novel global standard of care, atezolizumab and bevacizumab, the combination of a VEGF inhibitor, monoclonal antibody that we've used for ages, approved since 2004, and atezolizumab, a PD-L1 inhibitor. I will not go into a lot of details about this trial. You can look at the schema here. It's fairly straightforward for a phase 3 trial, although ambitious, as much as ambitious were the PD-1 inhibitor trial. So co-primary endpoints of OS and PFS, looking at superiority particularly. And I'm not going to bore you to death about the characteristics of the patient population, but just take one message home from this slide is that 73% of the patients had evidence of extrahepatic spread and macrovascular invasion, to suggest that these were patients with true advanced disease and with poor prognosis. I hope that will stick into your mind when you then look at the median overall survival that was achieved with this treatment, 19.2 months, something that when I first started doing HCC was absolutely inconceivable for a patient with metastatic disease with portal vein thrombosis and extrahepatic spread. And you can see that in this particular trial, sorafenib did particularly well, 13.4 months. And the secondary primary endpoint of PFS was also met with about a 2 months extension. Bit of a reflection point for me. I think that tiny little bit of increase in PFS does not mirror the wider effect on overall survival. So there is still something that we don't really capture in the way response translates into overall survival in this patient. And this is really the subject of ongoing research in these patients. The second thing that I think will please surgeons like Matteo is the fact that finally we are achieving a tumor response. We can actually see the tumor shrinking. And even in patients with heavily metastatic disease, you could see patients achieving a complete response and a partial response. Why I'm saying this is of surgical interest, because we are all having an appetite to combine these treatments to achieve higher cures. And I think potentially thinking about adjuvant, neoadjuvant therapies is really what is going to potentially give us more opportunities to attack these tumors at an earlier stage. And so this is, again, food for thought for the future. Safety, we know that, atezo-bev is a fairly safe treatment to give. However, it does have a number of complications. There is a risk of bleeding with bevacizumab. But if you look at most of the adverse events documented in the trial, a lot of them were actually fairly asymptomatic, like hypertension, proteinuria, liver enzymes increase, things that we are used to deal with as oncologists way better than things like diarrhea, PPE, and all these sort of interesting features of toxicity that we are all aware of with TKIs. Checkpoint doublets, so atezo-bev is one of the options, but not the only option, and we have opportunities to talk about the combination of PD-1 and CTLA-4. This was evaluated in a phase 1b study of nivolumab and ipilimumab, so the two combination of checkpoint inhibitors. You can see on the right-hand side that the response rates were actually quite good, approaching again 30%. And remember that this was a study of second-line patients, so people that already had sorafenib. Quickly looking at the Kaplan-Meier curves, you will see that the median overall survivals are actually very generous. And they're even better if you achieve a complete response. Look at that orange curve. So if the immunotherapy is working in you, like you have really good chances of being alive at 30, 36, 39 months, as we are seeing in the melanoma studies. Again, something that is unprecedented. And I think 2023 might be the year where we'll be seeing the randomized compartment of this study potentially being published, hopefully, or presented in one of the conferences. Something has already been made aware to the scientific and clinical community about the efficacy of CTLA-4 plus PD-L1. These are different molecules from AstraZeneca, but at the same time, the study called Himalaya, published this year, and where Lorenza was definitely part. So I'm really looking forward to have this conversation with her today. Is to basically combine, the CTLA-4 plus PD-1, and also have the opportunity of looking at PD-L1 alone compared against sorafenib. This is a mammoth trial of 1,300 patients that you can see actually managed to demonstrate something that is very, very interesting. First of all, that you can achieve long-term survivorship from a combination of CTLA-4 plus PD-L1. You can see 36 months overall survival, the curves that are detaching when durva-tremi are given together. And in this particular trial, the tremelimumab was only given once during the first cycle, and then the patients were maintained on one drug only. And the durvalumab given alone in absence of the tremelimumab also does, not too bad, with median overall survival about 16 months. And is certainly being declared not inferior to sorafenib. So I'm really looking forward to try this treatment and see which patients might be best for each and every one of them. The response rates are quite favorable, 20% in this particular combination. However, just to point out that the toxicity might be perhaps a little bit more of a problem. And in particular, I like to think about the grade 3 toxicity, which comes up here, as you can see. It's 50%, in particular treatment related, around 25% for the combination, which is nearly double the monotherapy. So this is something that we need to keep in mind when we're counseling these patients for treatment. Another avenue for treatment is the combination with PD-1 plus TKIs. And this particular study was one of the most exciting that was published back a couple of years ago now, looking at the role of pembrolizumab plus lenvatinib in first line. I think those waterfall plots talk for themselves. It's only a minority of patients that actually don't produce a significant response. The vast majority have some form of shrinkage or stability. However, we are then left into the dark because last year in September, the phase 3 counterpart of the study of pembrolizumab was actually declared as a negative study. And you can see on the right-hand side that the curves of the combination pembrolizumab are not better than lenvatinib alone. I must say, I don't know what Lorenza and what all of you think about this trial, but I still have to digest this data. But one thing is sure is the fact that there is no evidence of synergy between the two compounds, as you can see. On the other hand, in the camre-rivoceranib study, which is a similar study, the molecules are essentially the same, but trialed against sorafenib, you can see that the difference is very, very broad in both PFS and OS, which gives me the sense that these trials should be compared one against the other to say, well, sorafenib and lenvatinib are not really the same thing, and maybe we should really reflect on this for future studies. So novel combinations in first line, you can see that there are plenty, and I apologize for the fact that this slide is actually quite small. Certainly atezo-beva and potentially durva-tremi are characterized by a positive OS and PFS signal. Atezo-cabozantinib, another study where my friend Lorenza was heavily involved and led globally, unfortunately didn't lead to a positive signal. And as you can see, pembrolizumab and other TKI studies are at the moment a little bit of a question mark. First, because one didn't really prove superior to lenvatinib alone. Second, the Chinese-oriented study might not find replication in terms of validity in patients outside China unless further efforts are planned to run this study in non-Asian population. And we're also left with other options such as PD-1 monotherapy or TKI. I like to call this the Pinatogram of treatment options in HCC, and you can see it's getting more and more complicated as we go along. But as you can see, at least in patients with advanced HCC, immunotherapy has become an option and a standard of care, which competes with sorafenib and lenvatinib. And I have to say that in patients who are eligible for immunotherapy, at least in my clinical practice, atezo-bev should be considered as first option. We might at some point have durva-tremi fully available to us. We don't have it in the UK just yet, so I have got no experience of using these drugs as of yet. But I think there would be an interest in using these, especially in patients who might be contraindicated to use bevacizumab. I have no idea what will happen to Cambra-Revo, as we said, because at the moment there are no plans, as far as I'm aware, for an immediate approval in Europe and in America. But as you can see, it's actually quite important to say that in second and third line, we are really going off the charted pathway, and we don't really know what to offer these patients. And this is probably going to be an important wave of novel efforts in clinical research. So I hope I managed to conclude to convince you that checkpoint inhibitors are effective in a fraction of patients with HCC. The combination is probably the way forward, given that it has already become a standard of care. There are unfortunately no biomarkers to guide treatment decisions, and we should invest on these academically and from an industry perspective. And that as we stand, toxicity and clinical patient profile is what really guides us to empirical prescribing. And with that, I thank you ever so much for your kind attention.
Thank you very much, David, for this very clear and great presentation, as usual. Let's see if there is any question. Giuliano, I think.
My role is to ask questions. So thank you very much for this comprehensive talk. You know, I treated the first HCC patients with imatinib. If you go back, it's been published in Anti-Cancer Research. It was 2004, the publication. At that time, you know, imatinib was the first tyrosine kinase inhibitor, and it was also effective for leukemia, chronic leukemia, right? So if you just do in vitro study and you put your PD-1 antibody into the culture, cell culture, PBLs, and you put imatinib together or sorafenib, what do you think will happen?
Well, it's actually quite interesting, Giuliano, because these studies have been done, and actually they have been published in highly respected journal, Clinical Cancer Research. So there have been the group of Joseph Lovett and Augusto Villanueva, who are actually quite fond of doing translational mouse models. I think they did it during the development of this drug. So there is an elegant study of atezolizumab plus cabozantinib, which is the study that Lorenza led, and you know, the interesting thing is that in the mice, you can actually see a really good anti-tumor response. And I think this is really one of the factors that I can't really explain. So how are these mice replicating the human scenario? And I think one of the people in the audience previously said, you know, is it a matter of the gut microbiome, for instance? Like, you know, I'm just pulling one thing as opposed to many others.
May I stop you? I asked for the in vitro because, you know, the effect of PD-1 or PD-L1 antibody, checkpoints inhibitor, is based, the hypothesis is based on in vitro studies. So you really don't know what happens in vivo. When you inject the antibody, you get binding, in the liver, in the intestine, in the thyroid. You really, really don't know. And if you believe that there is a tumor lysis induced by activated cytotoxic T lymphocytes, you should get signs of that in the serum, and that you get tumor lysis. But you get the opposite. You get disappearance of the tumor and nothing else. I mean, besides side effects, which may be really very prominent. So that's why was my question. If you do that in vitro, you speak about combination of TK inhibitors and antibodies. If you check it in vitro, it would be interesting to see what happens. I mean, I think that imatinib blocks DNA synthesis, RNA synthesis. You get, depending upon the doses, you get killing of PBLs, peripheral blood lymphocytes. So sometimes oncologists, I like them to think, not only about combinations, just try, but
No, I completely agree. And I must say that in the vast majority of these combinations were tried also because of competitive advantage from an industry perspective. So, certain compounds might have been more appealing than others scientifically, but obviously, you're going to push for multi-million programs when you have enough patent left. And so, for instance, I think that, some of these compounds like sorafenib, we will never see in clinical trials anymore because the patent is gone. Lenvatinib, I think the patent is about to go anyway, because it's already like nearly 10 years of use. And so in a way, you either push the academics to do spontaneous studies, which is quite tough because where do you get the money? Or, you're left with these, I call them fast food combinations sometimes. And I hope, I'm resonating with your thought that, whatever you have, you just combine because it's fun.
I want you just to think about the host, the role of the host. Tumor growth is probably due not only, by independent, growing of cells, but tumor growth due to growth factors.
Yeah.
Okay. Tumor growth factor. This is also for Dr. Mario interesting. So you not only do in vitro cultures, but also take some serum from patients and put in and see what happens. Okay. So
Excellent point.
What's my suggestion? Okay, I stop here from my point of view.
Domenico?
Hi, David. Thanks a lot for your presentations. It was very nice. And I couldn't agree with you more as immunologist when you said that, like the variables among like the tumor microenvironment are so many about cells. Because we are really deriving a lot of immunotherapies without even knowing which kind of targets and immune checkpoints on different cells we are doing. So we are like, thanks also to Matteo, we are performing like this kind of study on a large set, 40, 45 HCC patients. And other things that we saw among like different immune checkpoints, there is like a huge heterogeneity between from patients to patient, according to the natural history of the disease. So if the patients have like, a recent disease or a chronic disease, like it's very different. And there are some cells, for example, NK cells compared to gamma delta or alpha beta, they have different kind of like exhaustions, and they are different kind of rescueness for like, their capacity to kill the tumor. And, we are like delivering all these cells without any knowledge of that. In certain points, when we talk about this personalized medicine, I think we be needed like, to increase the efficacy of these potent drugs to understand which kind of patients we have in front of us. And so it would, what do you think about that?
I think, Domenico, I'm in full agreement. And I feel it's a bit of a shame that, obviously the companies will run the studies with a financial and competitive edge, which is fine. I don't think I've seen, for instance, any biomarker paper being published by any of the failed studies, zero. So these samples might be somewhere. Where are they? God knows. And I think this is really more of a public call to say, look, if there is evidence of activity or not, you're kind of duty bound to still answer some questions. And if the company is not able to support it, then individual researcher, charities should approach the companies and have free use of the samples, as much as we now are forcing the companies to release the data after the primary research has been completed. So I don't want to be like this big anti-company person. I think it's very important that the pharmaceutical industry continues to obviously fund these very important programs. But I think it's crucial that we maintain an industry academic leadership of these research programs, because otherwise, you know, Domenico, we're left to pick up the pieces after, you know, either drugs fail or then rerun the same studies again, or doing observational studies, because the original studies never really answer those questions. And it takes another 5 to 10 years. And, you know, all I care about is to improve care of patients. I don't care about selling drugs at the end of the day. And so I think
at the end of the day, we are like, you know, shooting with a bazooka without, you know, and unknown targets. You know, maybe like, you know, a little gun would be like even more effective with like a precise target.
Exactly. And so the societal benefit of all this research is understanding more. And I think we should maintain that as academics, obviously, you know, maintain that focus. But I think you're absolutely right.
Thank you.
I think there is another question, Mr. Mod Hassam.
My name is Mohammed Azam Kayasif from Dubai. Thank you very much. We are now in the era of microbiome medicine. If we really know what is the cancer microbiome signature, especially in HCC, can we reshape the cancer microbiome signature to increase the effect of checkpoint inhibitor or not?
What an amazing question. Thank you very much for this, Mohammed Azam. I think it's very interesting because there are a lot of things that we don't yet know about the fact that not only there is an immune suppressive microenvironment from the liver per se, but also from the cirrhosis and from the gut-liver axis, which is in cirrhotic patients, as well as in patients with HCC, is already deranged. The way I studied this in some of the previous studies that, you know, involved Lorenza and others in this group was to look at the impact of antibiotics, for instance, and see whether the disruption of the gut microbiota by using antibiotics is actually detrimental to the prognosis of patients with liver cancer, having this idea of modulating the microbiome in mind. And we found that actually this is the case. We're actually publishing a sub-analysis of the Imperial 150 study looking at antibiotic use, and we have seen that even in those curves that you have seen before with really good overall survival, if you disrupt the microbiome with antibiotics, the survival gets less. I don't think I have a recipe right now, Mohammed Azam. I think it's difficult. We don't know what are the bacteria that we should modulate and how to, if it's just a probiotic, if we just need to eat yogurt, if we need to take antibiotics, that I don't know. I cannot give you a clinically translatable answer. But I think that is a gold mine of research that we should continue investing in.
Thank you very much, David.